Hey students, this is Dr. Stabio, and I would like to present to you Stabio's 1, 2, 3 punch, a super simple formula for writing a winner abstract. And really, I have to credit my PhD advisor, Dr. Cornwall, who spent a lot of time with me as a young PhD student in his lab teaching me how to write an abstract, either an abstract for a grant, an abstract for a conference, or an abstract for a paper. The formula is the same. And he said that every abstract should answer 3 questions and have a punch. So these are the 3 questions. So step 1, you need to answer the question, what's known? And when you do this, the first 2 words of your abstract in this first sentence are key. They neurally prime the reader for what's to come. So if your abstract, if your research is about autism, autism needs to be one of the first words of your abstract. However, if your abstract is about, or your research is about neural circuitry, and you use an autism model to study neural circuitry, the neural circuitry should be the first word in your abstract. And you want to spend, 1 or 2 sentences referring to the existing literature, painting a picture, which is why I've put this icon here, of what is the state of current knowledge. So 2 sentences about what's known. Step 2 is, what is the gap in knowledge? What is not known? What's the problem or controversy? What's the unmet need in your field? This is your narrative hook. Okay, so you want to use sentences like, this leaves an unsolved problem, or however, it's unknown if X modulates Y, or however, XYZ remains poorly understood. This is where you grab the reader. Okay, step 3, you want to answer the question, what are you doing to fill the gap? So you want to clearly state what you did. What gap in knowledge are you filling? What unmet need are you providing? This is where you come to the rescue. You want to state your long-term goal or overarching objective of the research. So for example, you can use phrases like, the goal of this project was to, and here, if appropriate, you should also include a hypothesis. Okay, and then 1, 2, 3, punch. This is where you give the punch. Who cares? What's the impact? Why should anyone fund you? Why should anyone read your paper? So this is, statements like, this is important because pharmaceutical treatments cannot be developed without understanding these key mechanisms, or this will lead to improved quality of life for X patients suffering with Y disease, right? And no matter what your area of research, whether it's educational, it's clinical research, it's bench research, there should be some punch. Now the punch doesn't always have to go at the end of number 3. Sometimes it goes at the very end of the abstract. Sometimes it goes in the fill the gap, but I like to put it in after the 3rd step. Okay, after you do 1, 2, 3 punch, what's known, what's the missing gap, what are you doing to fill the gap, and who cares, then you can really take off with your abstract. So you want to give a couple of methods using the KISS method. Keep it simple, stupid. You don't need to go into an exhaustive detail, but you want to give the reader an idea of what was done. Did you use mice or using humans? Was this behavioral, histological, electrophysiological? For example, you might use phrases like, blank method was used to determine blank. Like viral tract tracing and reporter mouse line was used of X was used to determine the retinal ganglion cells that innervate the dorsolateral geniculate of the thalamus, right? So this is like jam-packed. There's a lot of data here, but it's also rather simple. Like, I'm not going into every single step that I did, right? If human subjects or if animals were used, you need to make sure that you either have an animal protocol or an IRB protocol. And you want to have a statement of how data either were analyzed, if this is for a paper or a poster, or how they plan to be analyzed if this is for a grant. Okay, you must have results. If this is a grant, you don't have to have your results. If this is for an abstract to a conference, or this is for a paper you're publishing or a capstone report, you must have results. Even if they're preliminary, you don't want to say we expect this, right? Unless you're writing a grant, you want to say what your results are. You want to include your end values, your statistics. and then a conclusion. You want to carefully interpret the results. One of the most common mistakes I see students make is that they draw a conclusion that's not supported by the data. They might, if it's an educational study, they might say this shows that this tool, improves learning, when actually they didn't evaluate learning. They evaluated self-perceived learning, or they evaluated confidence levels in students or overall satisfaction, right? So you have to be accurate with what your results actually demonstrate. You can have, statements like this suggest X, Y, and Z. The data demonstrate that X, Y, and Z. You also want to include a one sentence on limitations and future recommendations. Now, this is a lot to fit in to a limited amount of space, and the abstract will take you the most amount of time of anything. Like it is very time consuming, and it is the thing that you should spend a lot of time perfecting. You want to give it to colleagues or people in your lab or fellow students to read and proofread. The last step before you turn an abstract in is to make the title. You think the title should come first, but really once you get your abstract done, that's when you really know what your title is all about. You want it to be concise with all the key info. You want to make sure your keyword is there. You don't want to use acronyms in the title. Your first author is the presenting author. Who's going to present this, or who is the main person doing the work? This is if you're sending an abstract for either your capstone report or for a conference. Then you have middle authors. These are the ones who helped with the project. And your last author is the senior author, who's typically the PI, the principal investigator. You want to include the affiliations of all your authors. Don't forget to spell check and grammar check before you submit. Okay, so what we're going to do next is look at some examples from different types of research and see how this formula works. So the first example is from some previous MHA students, Jensen Lemira and Charlotte Wilson, together with Noah and our wonderful Kaylee Orr, Dr. De La Rosa from the dental school, and Dr. Greeny, Tom Greeny from the dental school as well. And this is a project that was done and published in 2023. Here it says one of the most difficult, and actually this abstract, not all abstracts do this, but this one separates the sections, which is really nice. Some of them it's all blended, but this one it's separated. So one of the most difficult local anesthetic blocks to master in dentistry is the inferior alveolar nerve block. Historically, dental students have practiced local anesthesia on one another. And at CU, these practice sessions have been limited to one lab section. Okay, so this is what's known, right? This is difficult and there's limited time, right? This is the problem. Step 2. The predictability and confidence of students' inferior alveolar nerve block success have not been high. Step 3. This is how the project came to the rescue. Therefore, this is a great transition. Therefore, right, and there's the goal statement. The goal of the study, the objective of the study was to investigate the impact of a novel simulator, blah, 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 blah. And then there's a section on methods, right? And you can see that there's the number of students is listed, how it was divided, what was measured, self-reported confidence and injection accuracy. And then you can see that they have some results here with some statistics. And a conclusion statement, a self-reported confidence improved, right? And anesthetic success was achieved. And this is future direction. Further investigation is necessary to determine long-term impact. Very clean, excellent abstract. Let's look at another one. Here's another one, novel targets in deep brain stimulation for movement disorders. This is from Dr. Thompson's lab in collaboration with Kern and Ogeman and Baumgartner in the Department of Neurosurgery. Here is what's known. Neurosurgical treatment of movement disorders, primarily via DBS, is rapidly expanding, evolving field. Although conventional targets include the subthalamic nucleus, blah, 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 other targets have been explored. You'll notice right off the bat that there is a lot of preliminary data. They're talking about all of these other targets other than the subthalamic nucleus, right? It's not until we get all the way down here that it says, however, reports of outcomes utilizing these targets are scattered and disparate. That is step 2, the gap, the problem. And then how did they come to the rescue? In order to provide a comprehensive resource, we have summarized the existing literature surrounding these novel targets. And what's the impact, the punch? This will improve, actually it's not even included here. Come on, Dr. Thompson, you should have really given an impact of this. I guess it's kind of it's within the, it's in order to provide a comprehensive resource for researchers and clinicians alike, but really the impact is improving, surgical outcomes and clinical care of patients. But the point is, now you may be saying, where's the methods? Where's the results? This is a review paper. And so there's not, as much of that other section. There's really just the 1, 2, 3 punch. Okay, let's look at another one. Human iPS-derived retinal organoids develop robust Alzheimer's disease neuropathology. All right, so this is from Dr. Vergara's lab, who's in the Department of Ophthalmology. She's taken capstone students in the past. And she talks about Alzheimer's disease, right? So this is the step 1. So this is step 1. What's known? You can see there's 1, 2, 3 sentences. Okay? You can pause and read this if you like. This is step 2. What's the problem? Given the devastating effect on AD, new treatment modalities that can significantly alter disease course are urgently needed. Okay? This is an unmet need. Step 3. In this study, we've developed X, Y, and Z. Okay? And then you can see she goes into methods using immunofluorescence and histological staining, right? AD-RO to control ROs, which is human, right? They're using human retinal organoids, stem cell derived. And then you can see a little bit of the results here. And this is the impact. This is a new tool for drug screening, biomarker discovery, and pathophysiological studies. Wow. Right? So in this case, you put the punch at the very end of the abstract. And like I said, the punch can kind of go anywhere. Let's look at one more. Here is one on medical education. And this is the use of 3D printed models in medical education, randomized control trial comparing 3D prints versus cadaveric materials. Okay, this is Paul McMenamin at Monash University in Australia. They're leading the world in 3D printing of cadaveric models. Here is their, now in this case, they only have one sentence of preliminary information. 3-dimensional printing is an emerging technology capable of readily producing accurate anatomical models. However, that should have been a semicolon or a period, grammatical error there. Because however is not technically a conjunction. Reviewers should have caught that. However, evidence for the use of 3D prints in medical education remains limited. So this is the missing gap, step 2. How did they come to the rescue? A study was performed to assess the effectiveness, right? Then they take off, right? They give their methods, right? They give their results here. They give a conclusion. This finding suggests that blah, 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 blah. And, what's the impact? Benefits to anatomical learning and supports their use in ongoing evaluation to supplement cadaver-based curriculum, right? It's not a huge punch. It doesn't save the world, but it's very important to people who care about anatomical learning, right? So again, they put the punch at the very end here. So your homework now. So we have provided you with this abstract formula. This is the same content that's in the PowerPoint. And on the second page, your homework before you come to class is to prepare your answers for your project idea, your capstone project idea. What's your project's one word, right? So I had a student in the past who was looking at, whose capstone project was evaluating the use of virtual reality for surgical planning for pediatric heart valves. right? And her one word, everything at the beginning of every paragraph and every sentence was about heart valves. But I'm like, you're not really studying heart valves, you're studying virtual reality, but you're using it in heart valves. So they're related, but I said your project's one word is really about virtual reality, and that needs to come front and center. Because you don't want to lead your reader on. You don't want them thinking that the project is about something that it's not. Okay, you need to answer what's known, what's the missing gap or problem, and how did you, or in your case, because you're at the beginning of your capstone project, how did you or will you fill the gap? Okay, so please answer these questions, brainstorm this, and come prepared to share it in class for the next session.